Hello, everyone, and welcome to today's live broadcast, a pharmacogenomic approach to medical cannabis, presented by Jahan Marcu, Director and Chief Scientific Officer, Americans for Safe Access. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom and/or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Marcu. I will now turn the presentation over to him.
Hello. Thank you for tuning into this webinar on a pharmacogenomic approach to medical cannabis. So, as was mentioned, I work with Americans for Safe Access, which is a medical cannabis advocacy group. It was founded in 2002 with the mission of ensuring safe and legal access to medical cannabis for research and therapeutic purposes. ASA focuses on direct action, education of both lawmakers, doctors, and patients, as well as making policy, consumer safety, and research recommendations. I'd encourage you to check out safeaccessnow.org for a wealth of free reports and resources provided by the nonprofit, as well as our patient-focused certification program, which contains training and product certification information for medical cannabis products. Part of this presentation and some of these slides actually come from our educational materials, which are available on our patient-focused certification website. Today, we're going to cover the basics of the endocannabinoid system, or the ECS, some of the basics of administering cannabis products, how they're administered, what are some of the issues with administration, and how it interacts with the system called the ECS, or endocannabinoid system. We're going to talk about a field of research that I'm very interested in and have, I guess as part of the disclosure, been advising a pharmacogenomic company that is interested in reducing adverse events and drug-drug interactions with patients who are using medical cannabis products, both in their personal life and in research studies. And we'll discuss some of the variables in dosing and some fascinating research about why people may be responding very differently to THC. And lastly, I'll end with discussing some network capabilities and areas for research. There's a lot of interesting research going on right now and a lot of really fun institutions if you're interested for learning more about really harnessing the power of medical cannabis. So the endogenous cannabinoid system, or endocannabinoid system, was fairly recently established. In the early 90s, the receptors were discovered, which led to the foundation of the endocannabinoid system field. Now, before that, no one really knew quite how cannabis was working. In the 60s, THC was purified and isolated from the cannabis plant, both in the Czech Republic and at a university in Israel. And from there, they were able to create a lot of synthetic versions of it, radioactive versions, and trace where it went throughout the body. And they found specific protein sites called cannabinoid receptors. And we'll discuss those a little bit more in detail later. But why they thought it might make a good medicine is because the receptors are found nearly everywhere in a large abundance. Probably one of the most abundant proteins in the brain are the receptors for this system that also interact with THC. However, one thing that's really interesting about these receptors is they're found everywhere in the brain except the brainstem, and the brainstem controls respiration. And that's one of the reasons why there's been no report of a toxic lethal overdose of cannabis, is because of lack of those receptors in the brainstem. But they're found pretty much throughout the human brain and throughout the body. Whoops. So what does this system do? Well, it helps us to eat, sleep, relax, forget, and protect is the term that's been coined. Cannabinoids such as THC and the naturally made compounds like anandamide, they help us to basically stimulate our appetite. They help us to relax. They help us to sleep. They help control pain and inflammation. They also help regulate memory. They help us forget things, which is a very, very important thing. I live in New York and I'm in California now. And if I had remembered every face I saw in the last 2 days of travel, my head would explode. So it's really important to have a system that's functioning properly to get rid of information that's no longer useful or perhaps harmful. And I think this is a very fascinating area because of the potential research in mental health and post-traumatic stress disorder. This system is also a protective system. The compounds on the cannabis plant, while they can cause intoxication, they're non-toxic. They do not damage cells. They do not kill cells. And the system within our body actually actively works to make sure the body's in homeostasis, getting things back down to normal, and keeping things from getting too excited. So this system within our body is encoded by DNA. It generates the little machines that generate the natural THC-like compounds in our body that produce the cannabinoid receptors. And if this system is out of whack, or there's a mutation, as has been described in some human populations, this is thought to be part of a deficiency syndrome called clinical endocannabinoid deficiency syndrome, first described in 2004 by my colleague, Dr. Ethan Russo. Now, this is thought to underlie some treatment-resistant conditions. For example, in some populations of anorexia bulimia patients, there are mutations in the receptors for THC. And this makes a lot of conceptual sense. If the major receptors in your brain that help control and regulate appetite aren't functioning properly, you're not getting the signals telling you you're hungry, that may make it difficult to treat that condition, except perhaps with a cannabinoid, which would then sort of stimulate the system to its proper level. Also, what's very fascinating is, we live in a world surrounded by cannabinoids. THC and other famous compounds are only found on the cannabis plant, but several species of plants make cannabinoids. There's one we may discuss a little bit today that's a terpene that's commonly found in black pepper that is also a cannabinoid and stimulates receptors. If you eat a salad, you're going to eat about 200 milligrams of naturally occurring non-intoxicating cannabinoids. They're just there in the diet. And some of these are being studied in the clinic and in animal studies for their therapeutic uses. So when we're talking about producing cannabis as a medicine, I think it's worth understanding where it's coming from before we go into how it's administered. For those of you who are unfamiliar with plant-based medicine, on the left, we see a fairly healthy cannabis plant. And if we were to say, target this for extraction, say we want to purify a drug. And part of the reason I bring this up is because when this airs a couple weeks later, that we're expecting a decision or a recommendation by the FDA to reschedule cannabidiol, which is one of the primary cannabinoids on the plant, and to reschedule it for the treatment of epilepsy. And it is purified from a plant source. And this is important because some people are interested in why choose a plant source over synthetics. Won't there be trace amounts of THC and other things in the purified compound? Yes, but it's also true for synthetic drugs, which may have other things in them too, in trace amounts resulting from the synthesis process and the manufacturing process. But when it comes to cannabis, when we're talking about targeting it for extraction and the making of resin-containing products, whether it's for a THC, cannabidiol-rich product, we're not targeting the fan leaves. We're not targeting the stem, the roots. These contain little to no amounts of cannabinoids. The unfertilized flower tops, as the female plants produce oil or resin, if they do become fertilized, such as in a wild field of outdoor cannabis, the resin production drops dramatically, and because it's going into sort of seed production. Hemp seed does not make any cannabinoids. And it should also be noted that hemp is just a variation or a variety of cannabis. Lots and lots of it has to be grown because typically it's about 5% or less THC or CBD is produced on there. So lots of it has to be grown. But largely what we're targeting on plants are trichomes that produce the majority of these compounds, majority of the cannabinoids. The seeds and the seed sprouts contain 0 cannabinoids. Any THC found on hemp seeds or cannabis seeds is a result of contamination. And so we're talking about cannabis products. We're talking about targeting a very specific part, the flowering top of the plant, where these specialized trichomes are. So there's a number of ways to ingest the fruits of cannabis and the extraction of these trichomes. Perhaps the most common one since ancient times has been smoking it. And this, again, can be, while the most common route of the administration is the least advisable. And this is administered through cigarettes, water pipes. If you were wandering around North Africa 1,000 years ago, you might have used a stick or a bone. There's some very interesting artifacts throughout the world demonstrating people making various bowls and things to consume cannabis. It's definitely cannabis is part of ancient history, and probably so is the chronic coughs and respiratory issues with smoking it. Also, a complicated thing with cannabis is that in a lot of places in Europe, it's combined with tobacco. But this is an extremely wasteful way to consume the product. At least 30 to 50% is lost through side stream smoke that is not inhaled. Another maybe 30% is destroyed from setting it on fire through the smoking process. It is a very inefficient and wasteful process, and really no health professional advocates for smoking of any product. Luckily, there's vaporization has become an option. Vaporization, such as the cannabis up in the right-hand corner in that yellow pharmaceutical canister, that is a version of cannabis which meets pharmaceutical standards and is available in the pharmacies in Europe. In that form, a whole plant form, that would be a Schedule I drug with no accepted value. However, THC in solution is Schedule II. THC as a pill is Schedule III in this country. So if you're playing the home game, it's a little complicated sometimes to keep track of the 3 different scheduling categories that THC appears in. But vaporization has been one way to reduce the carbon monoxide and to safely deliver cannabis. But what we're talking about today with pharmacogenomics is oral administration. Since 1985, pure THC encapsulated in sesame oil has been available and has been used in a number of studies. Up above, you see dronabinol or marinol. It's a very expensive pharmaceutical medicine in those round little balls in 2.5, 5, and 10 milligram amounts. I like to refer to it as the caviar of prescription medicine, one, because of its appearance, and two, because it's very expensive. And so as we're talking today about pharmacokinetics and pharmacogenomics, we're largely going to be discussing cannabis in the form of dronabinol, not so much in the Schedule I form of a whole plant material. So part of the way cannabinoid receptors work is through G protein coupled receptors. So if you're into pharmacology, this might be interesting, but this is important to note because they belong to a super family of class A G proteins, which are among the first receptors of this type to evolve. CB1 and CB2 receptors are actually very closely related to rhodopsin, which evolved in the eye to detect light. And so basically what happens is on the outside of a cell, you have an agonist or drug which binds to a receptor and stimulates it. This active receptor releases a signal into the cell, amplifying. So it's all tightly bundled in a little G protein complex. Something like THC stimulates it, and it sends off its downstream cellular responses to go about their cellular business. And some of those things involve turning on and off various ion channels, activating various kinases and protein activity. But what this really essentially boils down to is that by inhibiting certain ion channels, by stimulating inwardly rectifying potassium channels, cannabinoid receptor stimulation, notably with CB1, results in a decrease in transmission of neurochemicals, such as glutamate or GABA. So if you want to impress your friends, you can say, oh, I know what THC does. It reduces the release of glutamate or GABA. If you have a friend who's in pre-med, they will be impressed. And this is basically how it works. If you're getting a bunch of signals from a presynaptic neuron on the right down to the postsynaptic, reducing the signal might be of a benefit. So there are two main receptors that have been discovered to date, CB1 and CB2. Now, in general, activation of CB1 results in an inhibition of neurotransmitter release. The type 2 receptor, this is primarily found throughout the immune system and in bone cells. We also have many other proteins throughout the body that these compounds interact with, such as GPR55, GPR18. There's also ion channels. And this is, again, once the drug enters the bloodstream, it's going off to the brain and other sites. It is stimulating these receptors, as well as some of these receptors on the gut. But what also plays a factor is how these drugs are metabolized. So first off, I think it's good when I mentioned earlier that there's a very high level expression of the type 1 receptor, the CB1 receptor. And it's really interesting because the distribution throughout the brain actually parallels pharmacology, things we can kind of see. The nucleus of the solitary tract, you target receptors in there, you're going to stimulate appetite. You have things like the hypothalamus and the amygdala, things that help control emotion, and fear and things like that. Motor systems, I sometimes think of jazz musicians and cannabis when I think of muscle memory and things like that. But also for certain types of neuropathic pain, this could make a lot of sense. In the eye, you find cannabinoid receptors, and this is important because that's one of the first indications for cannabis as a medicine was for glaucoma and decreasing intraocular pressure. We also find that cannabinoids can interact in the gut, the immune system, as well as abundantly expressed in some cancer cell lines, as well as in many other peripheral parts, heart, lung, adrenal, kidney, liver, colon. You name it, there's probably the machinery of the endocannabinoid system functioning in that area, except for the brain stem. Type 2 receptor is more of an immune receptor. It's abundantly expressed in bone cells, the cells that make and degrade bone. And what's fascinating about this as we transition to our discussion of the role of genetics on pharmacology, CB2 polymorphisms, so there's these subtle changes in the genetic code, are actually associated with osteoporosis, severe age-related osteoporosis in human populations. In mice that are genetically bred without these receptors, they have accelerated trabecular loss. This is still a bit controversial because a lot of the mice that are bred without cannabinoid receptors, CB1 or CB2 or CB1 and CB2, many of them die at birth from a failure to thrive. If you don't have cannabinoid receptors, it's hard to get signals that you're going to eat and things like that, and develop that food reward pathway that's so important. So there is some concern over that model. But in human populations, it's been observed in at least 3 distinct human populations. It's also found on microglia, the immune cells of the brain, the type 2 receptor. We've heard of gliomas, can be a very nasty cancer, and we find CB2 receptors expressed on those, making them a great target for inhibiting cancer growth, at least in laboratory models. So as far as the endocannabinoid system goes, there's been a number of reports in both animals and humans about various polymorphisms and mutations in the type 1 receptor, which in humans is encoded as CNR1, is the name of the gene for the CB1 receptor. And this is associated with mental health issues, substance use disorders, neurodegenerative disorders like Parkinson's, as well as working memory. And this is, again, a field that's just sort of taking off. But what I wanted to talk to you about was not so much the mutations in these receptors, which I think will also begin to play a role, but it's unclear how these changes affect, say, THC's interaction. And so there can be huge variability in THC dosing. For example, some people use very low doses for effective results. Others require higher doses, even for the same condition. And we can use other things, perhaps observe other things in your life where some people get very low doses of one thing, other people require higher doses. One of those things I'd say is probably coffee. I can drink coffee probably a couple hours before bed and I'm fine to go to sleep. I know other people just smell it or have a sip of it and they're up. They can't sleep for a day. So again, that comes down to genetics at some level. So when you're trying to figure out the right dose of a medicine such as THC, this may require dose titration, right? Starting low, trying to stay low, and sometimes taking higher amounts. But that can lead to side effects, as well as going too low can lead to ineffective therapy. And there are many factors behind this variability, and that's part of the reason I started with the slides about cannabinoid receptor mutations, is I wanted to put out that there are many, many variables when it comes to this. But one important factor is not just the receptors, but the liver enzymes that metabolize our drugs. And so when it comes to pharmacogenetics, matching the pharmacy with someone's gene, we've discussed variations in the endocannabinoid system, right? Mutations in the CB1 receptor, mutations in the CB2 receptor, as well as there's been reported mutations in the enzymes that synthesize and break down the naturally occurring endocannabinoids in the human body. What's also fascinating to me as a pharmacologist is there are variants of CB1 that have been produced in culture which do respond differently to different drugs in terms of having different preferences for different drugs, different strengths of interactions. But what I'd like to transition over is talking about pharmacogenomics, which is much more broad. And here we're talking about drug transporters and drug metabolizing enzymes. Now, there are some really important drug metabolizers, P-glycoprotein or ABCB1. These are very, very important for many, many types of drugs. There are also drug metabolizing enzymes, the cytochrome P450 family, and these are sometimes called CYPs. And these metabolize a wide variety of drugs. And there are many different CYP450 isoforms. There's CYP1A1, CYP2D6. One that THC utilizes is CYP2C9. And these, again, if you're putting a lot of drugs in your gut, there's going to be some competition here. And part of our concern, especially my concern, is that when you're dealing with older populations, they've seen statistics of people over 50 are on an average of 4 or 5 medications. Those medications can have adverse events associated with interactions with one another, and that could potentially be to interactions at these CYP enzymes. So there have been a few studies that have been conducted looking at this and taking into account some of the pharmacogenomic responses. Many of them are ongoing or have not been sort of started. But what's interesting about some of the ones that have been published is, for example, in the CB1 receptor, they noticed that there was some influence or trends on effects of the drug THC, such as certain IBS patients, irritable bowel syndrome patients, were more susceptible to the appetite-inducing effects of THC versus other groups. As well as, transporters of the drug can affect their efficacy. But if you're interested in learning more about this research, we have the trial code here and some little information here to find about these ongoing studies about the pharmacogenomics of THC and other cannabinoids. So the National Institutes of Health has described pharmacogenomics, which is abbreviated PGX, as the study of how genes affect a person's response to drugs. This relatively new field combines pharmacology and genomics to develop effective, safe medications and doses that will be tailored to a person's genetic makeup. And so in our little diagram up top on the left, titled drug treatment, we have our genetic polymorphisms, which are all mixed up in a population. All the people with one mutation don't all live in the same place. We're all spread out. So it can be hard to suss out, one drug versus the other. And so, here we're looking at these 3 populations, the blue, the yellow, the red. One population may respond very favorably to the drug, one may have no effect, and one may be very sensitive or susceptible to the drug. And what I mean by that is if we look over on the right, we see our average THC levels. And so the CYPs, there are 3, we each get a copy of things, one from our mother and one from our father. And if you have 2 kind of bad copies, that could be an issue. And so scientists are super creative in the way they name things. So they found 3 variations and they're named 1, 2, and 3. And so participants that had the 3 copy, basically this is a different, this is a less active CYP. So you might think of the 1 as sort of the more quicker metabolizer, less in the blood, right? the 1,3, you have one copy that's not so active and one copy that's very active. But what we can just see here is that when we're comparing the average levels to each other, those that had a copy of the relatively low level metabolizing or active enzyme had higher levels, three times higher average THC, which you might imagine if you had 3 people in the room and they each took the same amount of a drug but had different levels of it in their blood, that could lead to difficulties in getting a consistent effect in patient populations. And this study was published in the Journal of Clinical Pharmacology. Another interesting example of this is also, like I mentioned earlier, CYP2C9. Now, this is a drug metabolizing protein, metabolizes THC and many other commonly prescribed pharmaceuticals. With a single amino acid change, this can basically result in much slower clearance of THC. So the first one I showed you were relative blood levels. This is now a study that looked at clearance. How quickly are you removing it from your body? And the CYP2C9 variant was very poor at clearing this from the body. And this could have a number of implications for drug testing, for how long it's around in your body. And I mentioned earlier too about synthetic versus natural compounds. And with drugs, such as some that are metabolized in the kidneys, some synthetic drugs, which are very effective medicines, leave behind residues. And one of the things that sparked my interest in this field was meeting a patient who had had a pharmacogenomic test done independently of their physician and found out that the one medication they were on, they were an ultra rapid metabolizer of, and they were taking higher and higher and higher doses. But the metabolites leave behind a residue that had to be cleared from the body. But it was building up a lot of this residue it was a drug for arthritis, and would have basically ruined his kidneys had he continued taking this medication. He showed the results to his doctor, and the doctor actually recommended a different drug that would not go through the kidneys, and therefore these residues and byproducts of metabolism would not build up at such high levels. But that's a personal anecdote, but they're just talking about another commonly prescribed drug. But what's also could be interesting too is that if you have a CYP variant and you're taking a commonly prescribed pharmaceutical with THC orally, there can potentially be some competition, drug-drug interactions at and that can alter levels in the blood. And there is some research out there on THC and CBD being able to alter levels of other drugs when they're co-administered orally. But there are a lot of variables here. How you're consuming it. Mostly we've been talking about oral consumption today, but there's also a field of study which is fascinating called inhalation topography. This is how people inhale things, such as if we use a classic like electronic vaporizer, people hold the button down for different lengths of time. They take different breaths, different deep, versus shallow breaths. We have the genetic component we talked about. There's also disease and conditions, such as the type 2 receptor and the immune receptor we mentioned. That is actually triggered to express at higher levels in certain diseases, like certain types of cancer cells will express high levels of CB2 receptors. If you twist your ankle, you will be, your CB2 receptors will elevate to try and control that inflammation. More receptors means more targets for the drug. So again, disease, healthy versus sick, could potentially also be a relevant factor too. also your lifestyle and your diet. There is a field of nutrigenomics, which looks at the way that diet influences your body in terms of your response to drugs as well. And what is really fascinating is the compounds that THC mimic within your body, the endocannabinoids. Those are synthesized on demand. Unlike other neurotransmitters, they're not stored. They're stored on demand as needed to help regulate homeostasis with the body. And if you don't have a lot of omega-3s and omega-6s in your diet, there is evidence in animal models that this could make it more difficult for the endocannabinoid system to function. In fact, in rats that are deprived of omega-3s and omega-6s, many of their G protein receptors just simply don't function. But all the endocannabinoids are again synthesized directly from cell walls, which contain different types of lipids and fats. And so your diet can potentially influence your interaction with cannabis and other drugs as well. So we have a lot of factors here that can influence someone's response. I wanted to include just a little bit of data on the metabolism. When it comes to smoking cannabis, the bioavailability is about 25%. So when cannabis is inhaled, you get peak plasma levels in 6 to 10 minutes. With oral, you're looking at probably more like 1 to 3 hours for onset for peak plasma levels. But there's high intra and intergroup variability, and it's due to those many factors probably that I mentioned. What is fascinating, I think, about inhalation of cannabis is that the metabolites generally, in about 30 to 45 minutes, they sort of start to balance out with the inactive forms in the body. So you start to get this interesting profile of metabolites about 45 minutes after inhalation. And over on the right here, what I like to just point out is perhaps just this one slide on the top right. You see whole blood, you see plasma. And if you just look up and down the plasma levels, you will see these graphs with huge error bars. This isn't a controlled study. People being administered these cannabinoids, and there's huge variability. In fact, at the top graph on the right, plasma for THC, the variability is so huge, they actually left the top of the error bars off because it wouldn't fit nice and neat in this field. So you can only see the bottom error bars, and the range is, 100 to 50 nanograms, and we're not quite sure, but according to this graph, the variability would be quite huge. And I guess the last thing I sort of like to talk about, if you're interested in patient and doctor CME, cannabiscarecertification.org is a great site powered by Harvard University's The Answer page. If you're interested more in the medical cannabis industry and training and certification, Patient-Focused Certification is a nonprofit third party that helps to train people at different levels, as well as people working in laboratories. The International Cannabis and Cannabinoid Research Institute is a premier private contract research organization currently conducting clinical trials in Europe. Some of their data is free on their website. They also look at product safety research, which there's a fascinating product safety study on the contaminants in certain cannabis products that they've acquired in Europe and tested. And they're also, again, engaging in some clinical studies in wound healing. The International Research Center on Cannabis and Mental Health, which is based in New York, is engaging in some much needed work on looking at cannabis and wellness. And one company, just as an example, the one that I have advised, Navigator Genomics, we are looking at utilizing their technology to apply it in different levels, such as in different studies and seeing if it can improve outcomes, predictability in different studies. And so I'd encourage you guys, if you're interested in learning more, interested in collaborating, interested in knowing what's going on in research, interested in educating yourself, these are 5 fairly good organizations that have some free resources that are putting on events. I think that they do good stuff. Lastly, I'd like to say everything you've seen in this presentation is from a research study, and largely they come from these 5 main resources. One is a recent book chapter that Dr. Ethan Russo and myself put out called Cannabis Pharmacology, The Usual Suspects and a Few Promising Leads. Happy to send you a copy of this article. But some other really interesting articles are listed here. One that I want to draw your attention to on the bottom, and I don't get any money, I'm not an author of this book, but Roger Pertwee's Handbook of Cannabis by Oxford Press is such a useful resource for, just a kind of a quick review by book chapter if you want to get a quick update on cannabis, quick understanding of the foundational knowledge. It's very well written. It's actually one of the few books that I have an ebook of that I like so much I carry it actually on my phone so that I can reference things, quite useful. But these other studies are very interesting because they discuss combining drugs and how that affects their pharmacology, how that affects their subjective effects. So if you're interested in learning more about, well, what happens when we combine different cannabinoids, what happens when we combine different cannabinoids with different drugs, these are great, great resources to start. And a lot of them, I think, are now free and open access online. With that, I would like to say, thank you for your time and attention in this tour of cannabinoids and pharmacogenomics. And I'm happy to take any questions.
Thank you, Dr. Marcu, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, what does medical cannabis cost the average person?
This is a great question. And this is another reason why I believe knowing more about, pharmacogenomics and more about the endocannabinoid system can potentially help save patients money. I think most people would be shocked to learn that according to some of the marijuana business organizations and some of the statistics they've put out, that the average person who's consuming cannabis spends about $1,200 a month. That is between $800 and $2,000 a month on average on medical cannabis products. And again, this is not covered by insurance due to the Schedule I status. And so I really hope that in the future, pharmacogenomics can be used to help patients find a medicine that works the best for them, that is match a variety of cannabis or perhaps a particular product or a particular route of administration, help save them money and time. But yeah, the average amount, according to some research, is about $1,200 with a range of $800 to $2,000.
What do pharmacogenomic tests cost?
There's a range depending on all the bells and whistles, but they typically range between about $200 to $800. If they're very, very specific for one mutation, sometimes they can be covered actually by insurance, but largely if you're getting a whole panel of proteins tested, it's sort of non-specific, you just want to see everything. That is typically not covered by insurance, but I think it's worth a conversation with your physician if you're interested. They may or may not have the capabilities to do it. Many PGX companies provide additional resources and guides for interpreting results and sharing it with your healthcare professional. And while these tests are out there and they're between $199 or $200 and $800, I think it's always worth discussing the results with your physician before you start acting on that information.
And you focused on THC, but could this also apply to CBD?
Yes, and I largely focused on THC because it has perhaps the most notable effects, and CBD does not cause intoxication, but it is a concern. There have been a lot of clinical studies looking at potential drug-drug interactions and its metabolism in human populations, but it absolutely could apply to CBD, which when taken orally requires, in the clinical setting, very, very, very high doses. We're talking hundreds of milligrams for an adult, things like that.
All right, we have time for one last question. How can this help reduce adverse events from drug-to-drug interactions?
I didn't spend too much time on that, but basically the SIPs that we discussed, if drugs can be competing for those, drugs can alter the kinetics, how those things are behaving. For example, THC and warfarin actually, which is a blood medication, will actually cause warfarin to increase very high in the blood. And so that could potentially be an area where you're like, okay, well, I should engage in some precautions here and monitor this. But again, I think part of what you could do with this information is to look at if you're consuming a cannabis-based medicine, to look at the pharmaceutical drugs you're consuming and what SIPs are they interacting with? Are they overlapping in any way? Is one preventing the metabolism efficiently of the other? And that's something we even see with just THC and CBD interacting with each other in the liver. So I think that, this information can be helpful to potentially look at areas where you might be like, okay, there's no crossover here. I can just go with standard precautions, or maybe there's a potential interaction, or maybe there's something that's been defined in the literature. And so by knowing a little bit more about your genotype, it's just one more way, one more kind of arrow in the quiver for you to use in your war chest to keep you healthy. And so I think that the point is that by knowing what isoforms, what forms of SIP you have, what variations you have, can help guide you to what the appropriate medication or medicine might be. And I think that that is sort of the general pathway to sussing out those drug-drug interactions and avoiding adverse events. Also, a simple way to avoid DDIs is, without knowing your genetic code, is by looking at overlapping symptoms like sedation. If you're taking two things that are sedatives, chances are they'll increase sedation above what you might expect, or if they have very similar side effects too. So again, your genome, your genetics can help inform part of the story, but also there's just some basic things like if the medications you're taking all have very similar side effects, that could be a potential drug-drug interaction issue too.
All right. Well, I would like to once again thank Dr. Marcu for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.